Welcome back to the Rooted in Resilience podcast. And today we are so excited to be joined again by our friend Georgi Dinkov. And we're going to talk again about cancer metabolism, an update on Georgi's cancer studies, what insights he's gained on how we can return cells back to proper energy production and reverse cancer, and any other fun updates from Georgi. So hello again, Georgi. Welcome back to the US after your recent trip.
Thank you. Thanks for inviting me again.
What was the best thing that you ate in Bulgaria while you were gone?
They have this traditional thing, which is like a phyllo dough stuffed with feta cheese and eggs. So they mix the raw eggs with the feta cheese, and they stuff the phyllo dough with it, and then they bake it. And it's called banica. And a lot of countries in the Balkans have it, but basically that's like when you go to Bulgaria, you have to eat that, and it's usually consumed with yogurt on the side.
That's perfect.
Yeah, that was.
Is that a perfect food? You got a little carbs, you got some healthy fats.
You got eggs, you have cheese, which is casein protein.
Yeah. Wow.
And actually, if they do it properly, the old school banica has very little dough in it, so it's mostly protein basically and a few carbs. And then on the yogurt, a lot of people add sugar to it in Bulgaria, so it's basically protein, sugar, and a little bit of fat.
Yeah. Wow. Great. So where are your cancer research studies happening? Where are those labs? And that's currently ongoing. Is that right?
Yeah. It's been going on for the last 2.5, 3 years. A lot of failed experiments before I got to something that worked.
So I think before we dive into those studies and what recent outcomes you're seeing, I think it'd be helpful to give a brief recap of what the bioenergetic view of cancer is. And so I think that it's really relieving and freeing to understand this version and view of cancer, because then you can understand that there is hope in reversing it. And we don't have to take this kill mindset. Instead, it's more support. And so can you explain a little bit the bioenergetic view of how a cell is going from proper energy production to cancer metabolism?
Well, anything that stresses the cell basically interferes with the proper use of oxygen. So the cell, because it needs energy to survive, and I usually give the analogy with the car. So the mainstream view of cancer is like the mainstream view of the car. What kind of gas you put in your car and how well the engine works is not going to affect whether you get flat tires or like a broken windshield or a broken door or the transmission will fail. None of that really is affected by the fuel. The bioenergetic view is basically looking at the car as a system, and every single tiny part of the car, which is a cell, we're all made up of cells, nothing else, depends entirely on the production of energy, which the cells themselves do. So anything happens with the cell, it can usually be tracked down to insufficient production of energy through the proper pathway, which is the completely oxidizing the nutrients that we use through the use of oxygen. So anything that stresses the cell, but because that process is slow and a little bit cumbersome, anything that stresses the cell and increases the energetic requirements, such as, let's say, you're exerting yourself, you basically, if you exceed the ability of the cell to produce energy through oxidative phosphorylation, which is the proper mechanism, then the cell will switch to something called anaerobic glycolysis, which means it's going to produce energy very fast but waste a lot of the fuel just to meet your energetic requirements. And we see that basically if you're doing some kind of an exercise and you work until exhaustion, basically once you start getting that burn in your muscles, that means the cell has switched from the oxidative phosphorylation to producing energy through the inefficient pathway, which is the anaerobic glycolysis, which is going to waste a lot of the fuel because it's not being oxidized, it's being fermented. And in the process, you're producing lactic acid. So this process, basically when you're in a stress state and the cell is producing lactic acid because it's wasting the nutrients that you've given it, it's indistinguishable. The phenotype of the stress cell, though not cancerous yet, is indistinguishable from the cancerous cell type. In fact, the only thing different structurally is that cancer cells tend to have damaged DNA, a very high rate of mutations, and that's one of the reasons why mainstream medicine has said, see, that's a mutant cell. Basically, this cell is no longer has the same genome as the healthy cells in your body. So since the cell is now no longer technically part of your body, of the organism, it's going to do whatever is best for itself, which is grow and divide, right? And then in the end, it will kill you because it spreads also throughout the body, forming metastasis, right? So the only chance you have for survival is if you kill that mutant alien cell. It's no longer your cell. It came from you, but it became something alien and it needs to be killed. So the bioenergetic view says, no, that's not true, actually. Basically, there are multiple published cases of so-called spontaneous regression of cancer. Cancer disappears in people, without any treatment. And, basically the medicine says, well, maybe something happened with the immune system. The immune system somehow killed the cancer, and that's why it disappeared. But the bioenergetic view says, no, there's no such thing as a cancer cell. Basically, there's an extremely stressed cell, which we call cancerous. And basically, that cell has a very deranged metabolism. And once the cell spends sufficiently long time in that state of deranged metabolism, it's going to start performing structural changes inside of itself. And some of the things that it does is that basically starts to disintegrate its own mitochondria, starts to basically, it cannot repair DNA properly. So that can actually explain the mutations seen in the cancer cells, right? And then the bioenergetic view says, well, if you supply the cell with sufficient amount of energy, right, it restore its ability to synthesize energy properly through the oxidative phosphorylation, these things should disappear. And that's basically, there's plenty of evidence for that. Some of the original cancer researchers, such as Otto Warburg and Linus Pauling and Albert St. Georgi and Hans Selye, they all thought of cancer as a metabolic disease. They said, yes, something is happening in the cell and it switches from oxidative phosphorylation to so-called aerobic glycolysis, which means continues to waste the fuel, even though there's plenty of oxygen around. So, but at the time they didn't have the microscopy, they didn't have the advanced knowledge the current medicine does of the structure of the cell, so they didn't know what's causing it, right? But now we do. And basically, if you look at the cell, the cancer cell, you'll see that there is nothing, that's happening in that cell that is basically, irreversibly damaged. And the bioenergetic view says, okay, let's make sure that the cell gets a sufficient amount of energy, and let's see what happens. And then we look at the pathways that in a cancer cell are being blocked, and we try to unblock them and see what happens. So that's basically
quick recap. I think it's important to remember the body has to stay alive. And so it's not like we have one way to produce energy, because that wouldn't be advantageous for survival. So in our last interview, you guys should definitely check that out. We talked about a 6-lane highway.
Mm-hmm.
And so on one side, you've got the most optimal lane for energy production, and that's oxidative phosphorylation. And you take all of the energy metabolites that you consume through food, that's essentially electrons, and those go into the cell, into the mitochondria, and go all the way to the end electron acceptor, and that's oxygen. So that's oxidative phosphorylation, and that is the best way for the body to produce energy, because we get 36 to 38 ATP out of that. We also get beneficial carbon dioxide out of that. And so that's the fast lane highway. That's where we're going to feel our best. We're going to not have cancerous cells. However, various life events or dietary or lifestyle factors can come up and block those highway lanes, right? So then we can't really rely on that as much. So we've got to go down the pathways, the alternative pathways that the body's designed that will still allow us to produce energy, but not optimally, right? And so all the way
it's
on
like the
left,
emergency lane in the highway. So basically, the cancer metabolism is like all the five lanes are blocked. The ones that you go high speed, and you only have the emergency lane. So it's going to allow you to basically continue to travel towards destination, but at a very inefficient way, at a very much lower speed. A lot of times the emergency lanes have these special damages on the asphalt, so they prevent you from speeding, right? So those are alternative, though basically they're an analogy of the blocks, metabolic blocks that are seen in cancer as well. And all we're trying to do is unblock the 5 highways, right? And have you move from the emergency lane into the lanes that the cell used to use when it was healthy. That's all.
And the cancer metabolism, the energy metabolites, they're not going through the electron transport chain.
They're not.
Even though there's oxygen present. So you brought up exercise. So I think that's easy for people to recognize. You can be out of breath, right? And so you have to rely on glycolysis, really quick energy sometimes. And that's okay for temporary reasons. That's okay. But it becomes a problem when that's your 24/7 reliance for energy production. And so those metabolites aren't going through the mitochondria. You're just getting, I think it's significantly less ATP, and you're getting lactic acid as a byproduct. You're not getting beneficial CO2, and CO2 creates it's like the best antioxidant, the best safety mechanism that the body has. The higher the CO2, the better oxygen delivery. And so you're missing out on ATP, CO2, and then you're getting buildup of lactic acid and other byproducts that are serving as more blocks.
Exactly. So, and also, so I guess we have the 3-lane analogy. So the metabolism of people consists of glycolysis, Krebs cycle, electrotransport chain. So it's basically you know, in a healthy cell, you're going through all 3. But when something happens, like a life event, or you're overexerting yourself, or you have toxins in your food, or in your environment, or you're dealing with stressful people, that increases the energetic demands on the cell. So anything that makes you feel like you're pushing yourself, right? That means the cell needs more energy. So you need to increase the energetic production to meet the demands. In a healthy young organism, that can happen relatively easily. But with age, because these blocks happen with food, with stress, etc., and they build up as well, right? So when you reach a certain age or a certain health state, basically you can no longer meet the energetic demands by increasing the metabolic rate. So basically then the body says, okay, I cannot use lanes 2 and 3 anymore because they have various blocks on them. I'm going to use lane 1, which is only the glycolysis pathway. And that lane is never blocked. If that lane gets blocked, you die immediately. So basically, and that's already been confirmed in several clinical trials where they try to inhibit the excessive glycolysis seen in cancer. And if some of the drugs were, you know, sufficiently strong to block it to the point where cells started massively dying, and of course, you know, there's no way to distinguish between a cancer cell and a normal cell because there isn't any. So they ended up killing the entire organism. So the emergency lane of glycolysis is always available, and whenever you cannot produce the energy through lanes 2 and 3, actually all 3 together, then you're switching over to lane 1, which is the emergency lane, the glycolysis. And that's what we're seeing in cancer. It actually has a specific name called the Warburg effect. Warburg, Otto von Warburg, is the Nobel Prize winner who discovered a thing in the 1930s or in the 1940s. And he himself thought that something is happening in the cancer cell. It's not genetic, and it's causing the cell to behave in a metabolically deranged way. So he thought until the end of his life that cancer can be cured through metabolic therapies. It's just at the time it wasn't known exactly what's going on in the cancer cell and what pathways need to be affected. So now we know that we have excessive glycolysis. We have a blockage at an enzyme called pyruvate dehydrogenase, which links highway lane 1, which is the emergency lane, to highway lane 2, which is the Krebs cycle, right? And then we can also have blockages at various, you know, connecting lanes between lane 2 and 3, not lanes, but connecting links, which is the connection between the Krebs cycle and the electron transport chain. So if we look at a cancer cell, basically we have one or more of these links are blocked by something. You know, some of the blockages occur because there isn't enough of the required cofactors for these links to work. Pyruvate dehydrogenase is a crucial enzyme because that's the rate-limiting step for oxidizing glucose through all 3 lanes. So the first thing you see in cancer, actually, or in a cell about to become cancer and start spreading, is that you have a blockage of pyruvate dehydrogenase, which results in the accumulation of lactate. So there have been proposals probably like, you know, going back into the 60s and 70s of treating cancer with thiamine, which is vitamin B1, the main cofactor for pyruvate dehydrogenase. Needless to say, the pharmaceutical industry had no interest in that, but there are isolated case studies showing that there's benefit. In fact, there is this case study from China showing that this woman with very aggressive, I think, was osteosarcoma tumors that they almost never get cured. I think the survival rate of these people is like in the single digits over a 5-year period. Much worse than pancreatic cancer or melanoma or brain cancer. So when the doctors took thiamine, it created a solution and injected it directly into the tumor disappeared. So that we have corroboration that, you know, thiamine could potentially work. So what else is going on in the cancer cell? Well, the cancer cell is in a very highly reduced state because when you're only using the emergency lane 1, there's this cofactor which is needed throughout all 3 lanes. It's called NAD+, nicotinamide adenine dinucleotide, and that is its oxidized form, and it also has a reduced form which is NADH, nicotinamide adenine dinucleotide hydrogen. Basically, like it has a hydrogen attached to it. So that's the main carrier of the electrons from food to oxygen. So it's needed across virtually all steps of all steps that are linking the 3 lanes of the highway. So if you look at a cancer cell, because the NAD plus and the NADH, it's a pair that's a redox pair. It's basically one of them is oxidized, the other one is reduced. So as the NAD plus is accepting the electrons from food and carrying them towards oxygen, Eventually, through these oxidative processes, the NADH becomes oxidized back into NAD plus, so it can carry more electrons, while the electrons that the NADH carried were given to oxygen, right? So the byproduct would be water.
It's kind of like taxi drivers. So if you've got a bunch of people at an intersection that all need a taxi driver to get to their destination, and there's not enough taxi drivers, then you're going to have a huge buildup of people.
Yeah, exactly.
So you're going to have a buildup of these electrons, and that's reductive stress. And you need that NAD plus to move those people to the next highway lane.
Exactly. And what the body does, because it definitely needs to have these people removed from there because they may be hit by incoming traffic, which would be the analogy. It comes up with an emergency mechanism, which is basically it uses one of the byproducts of the lane one, which is glycolysis. It's called pyruvate. Pyruvate can also act as an oxidizing agent. Ideally, the oxidizing agent should be oxygen, but if it's not being used in the cancer cell because its usage only happens in the Krebs cycle and the electron transport chain, if that usage is not happening because these two lanes are blocked, then what do? I still need my taxi to be available to be capable of ferrying people around. So I'm going to empty the taxi. In other words, I'm going to oxidize NADH back into NAD plus using my emergency vehicle called pyruvate in this case instead of oxygen. But in the process, I'll create a very bad byproduct called lactate. And that's really what the cancer cell does. It says, okay, I cannot use oxygen. I need to shuttle these electrons around, right? I need to be able to produce energy to survive. Remember, no energy, cell dies immediately. So I'm going to produce energy in the only way possible that I currently have, which is glycolysis, because there's blockage in the other steps. And then I'm going to oxidize NADH back into NAD, and I'm going to keep that cycle going. But in the process, I'm going to be wasting the sugar that's feeding into glycolysis, getting converted to pyruvate. I'm going to waste it into lactate, and this lactate builds up. So for a long time, it was thought that lactate is just a benign metabolic byproduct waste. But recent studies have demonstrated that's very far from truth. It's now known as an oncometabolite. Not only it is associated with cancer, it promotes the growth of cancer because it's one of the most potent stimulators inside of the body of the formation of new blood vessels. So in order for new tissue to grow, it needs to be properly supplied with blood. And basically, the way the body handles this is increases the production of something called vascular endothelial growth factor, or VEGF, which is basically does exactly what it sounds. Basically, it produces new blood vessels. So drugs that inhibit this enzyme, VEGF, have been used for cancer for, I think, 30 years now. One of the first drugs, and it's still used for many cancers, called Avastin. It was one of the first lines of therapy for breast cancer. So basically, we now know that lactate is a bad thing. So we don't want lactate to build up. Well, it just so happens that if you administer thiamine, right, this enzyme pyruvate dehydrogenase starts to work, at least more than without it. So now you have less buildup of pyruvate and less buildup of lactate. In other words, the electrons from glucose are now starting to flow through the Krebs cycle and the electron transport chain. But another thing that I mentioned is also seen in the cancer cell is the excessive reduction, which means too much NADH, not enough NAD plus, and it's only NAD plus that can carry the electrons from food. So basically, so in other words, a healthy cell will have a preference for, more NAD plus than NADH. In other words, it's always capable of taking more electrons from food and converting them to energy. And this accumulation of NADH and reduction of lower NAD plus is seen in cancer cells because, like I said before, without the usage of oxygen, you cannot oxidize NADH except unless you use pyruvate, and then you build up of lactate. So we don't want that, right? So what else can we do? Can we supply more NAD plus to the cell? It turns out the answer is yes, by administering a precursor known as niacinamide, which is one of the forms of vitamin B3. It's a precursor to NAD plus. So basically, so if this would work,
I'm curious, what are the cofactors needed to make that conversion into NAD plus?
I believe one of them is copper, and another one I think is vitamin B2. So basically, you would need some other B vitamins as well, but there's rarely a deficiency of those unless the person is very old, has an iron toxicity.
So you guys are getting the what's happening in Georgie's brain as he's thinking about cancer metabolism and designing his cancer cells. studies.
So we look at everything that we know so far. Assuming the cancer is nothing but a metabolic derangement, right, which is happening because of multiple roadblocks. What can we do to remove the roadblocks, or at least push them aside so basically cars can continue to flow through the highway? And we said one roadblock was pyruvate dehydrogenase. So in other words, give it the cofactor, which is thiamine, right? So imagine this roadblock actually is movable, but in order to move, you need this specific driver called thiamine, right? So you give thiamine, and the thiamine can move the roadblock around and allow cars to pass through. Another driver, a very major one, is NAD+, because it's responsible for potential blocks not only at the pyruvate dehydrogenase. By the way, NAD+ is also required for pyruvate dehydrogenase. So there's two blocks there, right? And then also NAD+ is required for the Krebs cycle and the electron transport chain. So you have basically, it should be able to move blocks along lanes 2 and 3. So I said, okay, so if we cannot can we administer NAD+ directly? Yes, you can give niacinamide. Another way to do it, because I said that NAD+ is just the oxidized version of NADH, is to give something to the body that converts NADH back into NAD+. In other words, an oxidizing agent. Normally that's oxygen, but if we cannot use oxygen, what else can we do? Well, the body is an emergency uses pyruvate. We don't want that because the byproduct is lactate. Can we use something else that can accept the electrons from NADH and convert it back into NAD+? And the answer is yes. Various other oxidizing agents, commonly known as quinones. Methylene blue is one of them. The vitamin K family is another one. The tetracycline.
Vitamin K2 in cheese.
Yes, exactly. Yeah, vitamin K2, but it's not only in cheese. It's there are multiple. There's vitamin K1, which is mostly in green plants, but it doesn't really have much of a function in the body until it gets converted to vitamin K2. And the reason it's called K is because from German word Koagulation, which means coagulation. So it's the main coagulatory factor in the body. So you can give vitamin K as a quinone. You can give methylene blue. There's something called coenzyme Q10, which is a cofactor for electron transport chain number 2, complex number 2. It's also a quinone. It's a benzoquinone. In fact, multiple studies have already tried using coenzyme Q10 for multiple diseases. And some of them are very successful, which kind of tells you that metabolic, maybe all diseases are metabolic. It's not just cancer. Because they're so diverse that if one thing worked on so many of them, then it's got to be underlying principle that unites all of these diseases, despite what medicine says. There's a clinical trial for Parkinson's disease. There's a clinical trial for Alzheimer's disease. There's a clinical trial for Huntington's disease, for multiple sclerosis, for inflammatory bowel disease, for irritable bowel syndrome. And not all of them, but some of them have shown great benefit. So there's something going on there. And the one role that coenzyme Q10 has in the body is acting as an electron acceptor, in other words, as an oxidizing agent. And it can do that not only at the complex 2 where it normally works, but just generically throughout the cell, so wherever it's needed. In other words, wherever you have too much buildup of reduction, which is NADH, right, or lactate, right, if you give more oxidizing agents, you should be able to convert NADH back into NAD+. And by the way, the enzyme that uses pyruvate and produces lactate is bidirectional. If you provide it with enough NAD+, it can take lactate, convert it back into pyruvate. So as a confirmation, there's several trials with humans showing that if you administer like 1 or 2 grams niacinamide daily, there's a drastic drop in the levels of lactate in the blood. So that corroborates our idea that basically giving a precursor to NAD+, it allows the body to use pyruvate as a normal source of energy and not as an emergency oxidant as what we're seeing in a cancer cell. Not only cancer, any stress cell will do that because, like we said, anything that stresses the cell, lanes 2 and 3 are blocked, you only get the emergency lane, right? So I said, okay, so we have thiamine, right? We have niacinamide. Let's see what they can do for cancer. So I started doing experiments with mice, but the tumor that is implanted on the mice is a human tumor. And it's a pretty decent model of the human organism. Of course, ideally it should be a human. So it's an animal host, but human tumor. Basically the first study I did was only with thiamine. So there was a slight benefit, but the mice still died, right? Then I said, okay, let's try niacinamide on its own. More benefit than thiamine, but still the animals died. Then I said, let's combine the two together. So basically the curve, the growth of the tumor started to flatten. The mice still died, but they survived much longer than anything the lab had seen for that type of tumor. So I deliberately chose the deadliest most treatment-resistant tumor that the lab had, because I said, if I can prove that this works for this tumor, then, it should be easier to accept that it can work for any other tumor. And the tumor that the lab proposed was something called human mantle cell lymphoma. And it's very difficult to treat. And when it's in that animal model, because the host is animal, basically it has 0% spontaneous aggression, never disappears on its own if you don't treat it. It has 100% mortality, and it's highly resistant to treatment. There's no known cases of cure in an animal model with this human tumor. So they said, okay, let's go with it. So thiamine by itself, a little benefit. Niacinamide by itself, more benefit. Together, the curve started to flatten. And I said, but they still died. They lived long enough, but they still died. So I said, okay, what else can we do? And I remembered that there was a recent clinical study with multiple sclerosis patients, specifically the most aggressive form of the disease, known as primary progressive multiple sclerosis. So typically, the multiple, I have a relative, but who has that condition, but the close relative. But the condition is basically the remittent relapsant form. In other words, you have periods of exacerbation, and then you have periods of recovery, followed by periods of exacerbation, periods of recovery. That's it's not a good disease to get, but it's if you have to get that one, then that's the least dangerous form of it. There's also secondary progressive and the primary progressive, which is basically these people invariably progress. They're usually in a wheelchair within 2 to 3 years after diagnosis. Nothing stops that condition, right? So, but then this human trial administered as a single therapy. Nothing else was given. No steroids, no other drug. The B vitamin known as biotin, or also known as vitamin B7. And then there was a striking amelioration of the symptoms for a disease that has basically nothing can stop it, right? And the proposal of the researchers was that biotin was acting as a stimulator of basically the oxidation of glucose. In other words, removing again the block from the pyruvate step, and then converting pyruvate, pushing pyruvate through the Krebs cycle in the electron transport chain. And then I found other studies, both in vitro and in vivo, demonstrating that administration of relatively high doses of biotin also drops lactate, right? And has a very good effect on diabetes, another condition that's also actually officially known as metabolic. People with diabetes also have high lactate. They don't oxidize glucose properly. In other words, they're also stuck in the emergency lane, but not entirely, like cancer. So you can think of cancer there's the spectrum of diseases. On one side is the healthy cell performing almost entirely things through oxidative phosphorylation, which means all 3 lanes are open and basically the food is speeding through them. And then in the middle of the spectrum is probably something like diabetes type 2. And at the very end of the spectrum is something like cancer. But if you look at the underlying derangements, they're all metabolic. I mean, everything structural that you do see, if you ever see it, it's only as a result of the deranged metabolism, because unlike a car, basically, if the car breaks down, you have to take it to the dealer. That's a structural problem. But in the cells, we now know that the cell has a self-repair mechanism. That's how it evolved. And all that it needs for that is sufficient, I call it currency, enough money, which in the cell world is actually energy, right? And then all you have to do, that's the bioenergetic view, but also corroborated through over 100 years of experiments, all the cell needs to do to repair itself is sufficient energy. So either give it the energy directly, and by the way, we can talk about it. There's a study with giving directly ATP infusions to mice with cancer. The tumors also disappear.
Amazing.
So another corroboration that it's energetic deficient that's really driving the tumor. And I had discussions with some of the doctors saying, yeah, at the doses you administer these things, they actually work like traditional chemotherapy. They're killing the tumor. No, they're not. We actually did tests on the animals. First of all, the LD50, which is the lethal dose for all of these substances that I'm using, it's for a human, it's probably somewhere around 20, 30 grams for vitamin B1. I'm administering the equivalent of 1 gram to 1.5 grams, the human equivalent. For niacinamide, probably around the same, about the same range. And more importantly, we did cytotoxicity tests on the animals when they expired. There was no cytotoxicity. Remember, chemotherapy, radiation, they all work through cytotoxicity. That is their official mechanism of action, because medicine says you need to kill that cell. If you don't kill the cancer cell, it'll kill you. So every therapy, except for the last few years when they're starting to move in the metabolic direction through something called immunotherapy, But even today, most of the traditional therapies for cancer are cytotoxic. So we looked at the cytotoxicity effects of these drugs that I'm administering. There was none. And it's not expected to be any because they're administered in doses that have been administered to humans for various other conditions, and there's been no issue. So, for example, why did I choose 1 to 1.5 grams for the that's the human equivalent for the mouse study? Because that dose has been given to people with Crohn's disease and multiple sclerosis and abolishes their fatigue with zero side effects. So it's not cytotoxic to humans at all. And then I chose the equivalent of 30 milligrams per kilogram niacinamide, which is the equivalent of 2 to 3 grams niacinamide daily for a human. Why did I choose that dosage? Well, because first of all, it happens to be the dosage which has never demonstrated any side effects in humans. And second, because there was the study that I mentioned when niacinamide lowered lactate, that's the dose that they gave to the person. I think it was 1 gram 2 or 3 times daily. So that was the therapy so far. And then biotin, I looked at the dosage that they gave the humans, where there was a drastic improvement in their primary progressive multiple sclerosis, and it was the equivalent of about 200 to 300 milligrams daily for an adult. That's a very high dose. And I was concerned that it would be cytotoxic. But if you look at the animal studies, converted into animal doses, there are multiple animal studies demonstrating you can administer, very, very high doses of biotin, the equivalent of several grams per day, without much ill effect. And then, based on the human study and the in vitro studies and the animal studies, I came up with a dosage that was around 2 milligrams per kilogram daily, which means for most humans, that means between 150 and 200 milligrams daily. And by the way, the human trial with multiple sclerosis used 300 milligrams daily with no side effects. So I used something lower than that. And as soon as I added biotin to the mix, the B1, B3, and B7, the tumor not only immediately started to flatten compared to the control group, but actually started to regress. So that was the first, and it regressed, but then it reached the plateau much lower than the original size, right? It reached the plateau and stayed there. So we kept the animals alive for several months. We kept them alive, but there was no regression and there was no remission. There was no growth of the tumor and there was no further regression. So we terminated the study. And I said, well, okay, good. So far, so good, right? And by the way, keep in mind, the trials between starting with thiamine and reaching the biotin probably did 7 or 8 studies were basically like trying different dosages because, I had to see what dosage works best. So maybe 7 or 8 studies that are encouraging but failure. And now we have the biotin, which now the tumor is starting to regress. So it's flattening, it's reaching a certain plateau, which at that level, by the way, for a human case, it will be considered a success because medicine is not trying to cure your cancer. It's trying to convert it into a chronic, manageable disease so that you're a client for life. Many people say, well, that's just a conspiracy theory, Georgi. No, it's not. So there was a company, I'm blanking on the name, but they came up with a cure for hepatitis C. And they priced it like relatively steeply. I think it was like $80,000 to $100,000 per treatment. But of course, the insurance is going to cover most of that. And as soon as they did that, there was the chief analyst of Goldman Sachs who was responsible for medical analysis because they invest in, obviously medical companies as well, came out with a blistering statement, said, short that company stock right now. That's the dumbest thing a medical company can do. And that analysis is on Forbes. I can send it to whoever wants it to. He said, the dumbest thing a medical company can do is kill its clients by curing them. So he's like, from a financial point of view, this makes absolutely no sense. So as soon as I started sending the article around to the people accusing me of conspiracy, some of whom are doctors, silence. So I said, okay, so we have 3 vitamins. We have a regression of a tumor, but not complete. What else can we do? And then, of course, I turned to our old friend aspirin. So if you look at aspirin, there are potentially thousands of studies in animals showing prevention of cancer, potential regression, et cetera, but several studies have shown complete disappearance of very dangerous tumors in animals at a human dosages that are in the range of just a few grams, 1 to 3 grams. So I said, let's try it out. And of course, we know Dr. Pete's theory. He's been writing about aspirin and cancer. He has an entire article about, I think it's called Aspirin Brain Cancer. And then I said, okay, let's try that out. So I tried it on its own at the dosage of about the same as the thiamine. The dosage was exactly as the thiamine, which is 15 milligrams per kilogram daily, which means between 1 and 1.5 grams for most people. So on its own, it flattened the tumor, but there was no regression. So then I added it to the vitamin combo. So it was vitamin B1, B3, B7, and aspirin. And lo and behold, within basically, and I think I've posted a graph on Twitter already, basically the tumors of all three animals completely regressed, and there was no recurrence. I specifically asked the lab to stop treatment and see what happens, and they stopped. There was no recurrence of the tumor. So then I started getting these attacks, quote unquote, by some of them, by doctors saying, well, of course it's going to cure the cancer, Georgi. We've known this for a long time. I said, really? Somehow I don't see anything in the literature about using B vitamins and aspirin for cancer. Well, maybe it's not published, but we know between ourselves that these are very toxic doses. They're going to kill the tumor. It's like traditional chemotherapy. So that's when I did the test for cytotoxicity. None. And by the way, those are lies. And the dosages that are used converted to humans, none of these treatments is cytotoxic. I already gave an example. The dosage used in my cancer experiments is the same dosage used without side effects for Crohn's disease and multiple sclerosis fatigue. The dosage for niacinamide that was used on humans many times. In fact, now they have a cancer study with the equivalent of 6 grams daily. I'm using less than half that, 2 to 3 grams for most people, no side effects. There's also biotin, okay? There's a study with humans using 300 milligrams biotin daily, no side effects. I'm using less than that, 150 to 200, right? And then for aspirin, older people probably know that back in the day, if you had rheumatoid arthritis or multiple sclerosis or like psoriasis or any kind of autoimmune condition, aside from the steroids, the only other treatment that they could offer was high-dose aspirin. And what do I mean by high dose? 8 to 10 grams daily in divided doses. Now that's high. My dose is the equivalent of 3 to 4 tablets. And it's basically, there's also actually a study using the exact same dosage, 2 tablets, 2 times daily, so 4 regular sized tablets total daily, which is pretty much the exact dose that I used for the mouse study with the human tumor. It was tried for relieving fatigue in multiple sclerosis, so it worked as well. Why am I so interested in multiple sclerosis? I have a relative that has the condition. So anyways, but it's just the dosage is extracted from human trials mostly, and it corroborated with animal trials as well. So none of these things that I'm using has ever been shown to have toxicity in humans at the dosages that I'm using.
Real quick, I think it's important to point out the obvious here is the bioenergetic view. Georgi's goal here is we're not killing. He's showing
Exactly.
this. There's no cytotoxicity. Simply the goal is to just remove those blocks and allow the cell to return back to proper oxidative phosphorylation, which this is something we touched on the last podcast, is proper glucose metabolism. So you can consider a cell it's not metabolizing glucose well, which is just so funny because you hear mainstream and all these alternative health camps still saying that sugar feeds cancer cells.
No.
And that's literally not happening because the sugar isn't going through to oxygen and the cell's not using sugar properly.
It's wasting it. So suppose the cell needs a specific amount of sugar daily in order to it's used not only for energy, but for synthesizing various intermediates that are very important for the body. For example, the D-ribose, that's a sugar that's used in DNA synthesis. It can be synthesized from glucose. So in other words, in order to build and repair, keyword, DNA, you need glucose. So if something is happening and it's preventing the proper metabolism of glucose, the way the cell overcompensates is by increasing its uptake of glucose from the blood. And that's exactly what's happening in a cancer cell. And it's being used as as an excuse by mainstream medicine to say sugar feeds cancer. But it's like saying if they have a very severe fire and you have an overabundance of firefighters, it's the firefighters that are causing the fire, right? No, they're there to help. I mean, they may not do their best job if the fire is too big, but you're not going to do it. You're not going to do any good by having fewer firefighters.
Yeah, it allows people to adopt this kill mindset.
Yeah. The cell is, the main thing here is the following. You did not evolve a mechanism to cure yourself. This evolutionary makes zero sense. Zero. There are things that the cells can do depending on the environment. And if you stress them, if you abuse them, the cells will try to repair themselves. But in order to repair themselves, they need energy. And if the things that got you into the state are still there and preventing the cell from synthesizing energy properly by using oxygen, using all 3 lanes of the highway, then the cell will synthesize energy through an emergency pathway. And by doing that, then the cell also says, well, this energetic machinery, this internal combustion engine that I have here is very elaborate, but I'm not using it. But it takes resources to maintain. So I'm going to start to dismantle it. And that's why in very advanced stages of cancer, you will see that they have very few mitochondria left. Some of the electron transport chain complexes 1 and 2 have also, almost completely been dismantled. And medicine is using that to say, you see, there is a structural defect of cancer. But it's not, that defect is caused by the lack of energy. The cell says, I'm going to, if that's my only lane available going forward, then I don't need the other lanes. I'm just going to destroy them. I mean, because it takes, so think of it of the cell also continuously maintaining the health of the lanes, I mean, filling holes and whatnot. And, you know.
So there it is. Like cancer prevention is maintaining proper energy production. That is literally, that is the key. Cancer
cure, or at least keeping it from killing you, is the exact same thing. Yeah. And it applies to many other diseases. I mentioned coenzyme Q10, already been found to be beneficial for many conditions. Its one role in the body is helping oxidative phosphorylation at a very specific step. But because it's a quinone, it may be able to help in other steps as well. We just don't know about it yet. But because its one known role is pro-energetic, then I would say it's a safer assumption to assume that many diseases, perhaps all, and especially cancer, are energetic in origin and not structural.
I do want to point out one thing. I think that all these supplements like thiamine, vitamin B3, aspirin, I think that biotin, I think that in the bioenergetic space and many other health spheres, like they're discussed how beneficial they are. And like, there's no getting around, those are important nutrients, but we are talking about cancer here. And I think people expect that they can utilize these supplements in a way to like that's needed for day to day. And it kind of puts less reliance on daily food, like what they're consuming day to day. And we do get a lot of these nutrients when we're eating well, when we're eating superfoods regularly, when we're eliminating foods that are causing more blocks in the highway. And so I don't want people to have the mindset of like, oh, I have to take these supplements to prevent cancer. We are talking about the reversal of a very serious state.
Yeah.
And so again, just stressing supplements can be very beneficial, but they're not going, I think a lot of people put too much reliance on them for day to day, whereas instead we should maybe be prioritizing cancer prevention with proper energy production, what we're eating every single day.
Exactly. I mean, all these things, they're dietary, right? Even aspirin, you could get it from willow bark.
Yeah.
At least its metabolite salicylic acid, which is responsible for about 80% of the effects of aspirin. So ideally, in a society that has not had its food supply compromised as much as Western societies have, unfortunately.
That's fair.
Yeah, then you probably won't need any of that. And in fact, if you look at we now call them primitive societies, but they're not primitive at all. Because if we look at health span and lifespan, they dwarf us. They almost never develop disease. I mean, they develop other disease, but usually infectious disease is what kills them. But if you remove infectious disease, these people almost never have cardiovascular disease. They almost never have cancer. There's a tribe that lives in the Amazonian jungle. I think they call the Tsimane tribe. They basically, their diet is entirely natural. It's about 80% carbohydrates, so very high sugar diet. They never have cardiovascular disease. They never have cancer. I mean, in fact, the cases there are so rare that they usually published in that Brazilian Journal of Cancer whenever they're found. So the incidence of cancer and cardiovascular disease in that tribe is like 1 in 100,000. While in the Western population, now the estimates is basically the lifetime cancer expectancy is 50%. So 1 in 2 people will get cancer throughout their lifetime. Same with cardiovascular disease. Diabetes is probably even higher.
So we really need this research because it can be reversed. So you've done B1, B3, biotin, and aspirin.
And aspirin, yeah.
And have you done any updated studies?
Yes. So why did I aspirin? So there's a lot of studies on aspirin, but not much knowledge about its mechanism of action. And I know, thank you for mentioning the glucose metabolism, because the reason I had aspirin is because it plays a very important role in something called the Rendell cycle. So the Rendell cycle stipulates, and it's been proven experimentally, that at any point in time, the cell can oxidize only one of the 2 macronutrients, either glucose or fatty acids. Some people say, well, we can also oxidize protein, but only after converting it to glucose first. So really, at the end of the day, the cell can only oxidize either fatty acids or glucose. So whenever the cell is stressed, you release these stress hormones. The organism is stressed, really. When you perceive stress from the environment, you release the two stress hormones, cortisol and adrenaline. And what these two do is basically cortisol starts to shred your lean tissues, starting with the muscles. And basically these amino acids go to the liver, get converted to glucose. Very bad situation because you don't want to lose muscle mass. In fact, a rapid loss of muscle mass is one of the most reliable predictors of future mortality. It doesn't matter if you have any disease or not. In the elderly, it's known as sarcopenia. Actually, not only in the elderly, but it's called, basically the muscle loss is called sarcopenia. And sarcopenia is perhaps the best predictor outside of the cortisol to DHEA ratio. So there you go, stress plays a role there again. And also serum levels of albumin, which is the protein produced by the liver to carry a variety of nutrients around. So anyway, so the cortisol and adrenaline get released as a result of the perceived stress. Cortisol destroys your lean muscle tissue and bones and skin and anything that's composed of amino acids, while adrenaline shreds your fatty tissue, and basically that fat from your fatty tissue starts to float into the bloodstream and the levels rise. And because for a lot of people the fatty tissue is overabundant, you get an excess or relative excess of fatty acids versus glucose into the bloodstream, especially if you're restricting sugar. That's why restricting sugar is so bad, because the body needs it. And if you don't give it, it's going to get it from somewhere. And you can think of your protein. Actually, you can think of your entire composition outside of your fatty tissue as basically composed of protein, which can eventually be, and is in advanced cases of cancer, is used for converting into glucose. That's why advanced cancer state patients have this gaunt look. It's also known as cachexia. In other words, a wasting process, because their stress levels, the levels of the stress hormones are very high.
We've been having this podcast conversation with Kathleen talking about the Minnesota starvation experiment.
Mm-hmm.
And one thing that study talks about is how the liver can shrink by 50% due to just like the body utilizing those protein structures.
Yep. Yeah. Anything in your body that is not composed of fat is composed of protein. So outside of the glycogen, which is a small amount, maybe half a pound stored in your liver and a little bit in the muscles. But that's a tiny amount compared to entire body mass. So the entire body mass, including the bones, right? Bones are 30% collagen. Yes, there's calcium there, right? And other minerals, but they're 30% collagen. So, and by the way, having high levels of cortisol has been shown to thin bone and make it brittle as well. Long-term usage of glucocorticoids, synthetic ones, which are often given for autoimmune conditions such as multiple sclerosis, they cause osteopenia and osteoporosis. So anyway, so whenever the organism is stressed, it releases stress hormones, which is natural. But the idea is that this period should be brief. You don't have an animal in nature that is constantly stressed 24/7. It wasn't meant to be this way. And if it is, the animal usually dies, not very long after. But in our Western developed societies, we have this very pervasive and pernicious amount of stressors that are around us 24/7, if you live in the city specifically. And they're very low, they're low grade. So you get like low grade chronic uninterrupted stress. And what this leads to is basically overabundance of fatty acids into the blood relative to glucose. And if you're not consuming sufficient amount of carbohydrates, then basically the cell will get predominantly fatty acids. Whenever you get predominantly fatty acids to the cell can only oxidize glucose or fatty acids, and it depends entirely on the external environment, which means the bloodstream, for supply of nutrients. So if the bloodstream is supplying mostly fat, then the cell will oxidize mostly fat. The problem with oxidizing mostly fat is that it blocks the oxidation of glucose precisely at the pyruvate dehydrogenase step. So if you have too much fat oxidation going on, administering the cofactors such as thiamine, niacinamide, and biotin, which can actually bypass the step of pyruvate dehydrogenase, may not be enough because the cell is stuck at oxidizing fat.
And isn't so we talked earlier about the NAD plus to NADH ratio. We want more NAD plus. We want more taxi drivers to bring the electrons through. Fat oxidation does not lead to... It
depletes. It lowers the ratio of NAD plus to NADH. In other words, it depletes the number of taxis you have available. So in a very real sense, it acts like a stress. We said, remember when the cell is stressed, especially in cancer, you have a reductive state. In other words, an overabundance of NADH and a deficiency of NAD plus. That's exactly what happens when you're over-oxidizing fat relative to
Is
glucose.
there a difference with PUFA versus saturated there?
There is, because PUFA can also break down into various pro-inflammatory products, either oxidatively, either enzymatically or non-enzymatically. Enzymatically, PUFA can convert into prostaglandins, the leukotrienes, the prostacyclins, and the thromboxanes, all of which are inflammatory, all of which.
Just destroy your DNA and other structural stuff inside
Just
your cell
cause systemic inflammation. And in fact, drugs that inhibit some of those enzymes that use PUFA as a raw material are widely used clinically. They're known as COX inhibitors. By the way, aspirin is a COX inhibitor. So right there and then, and by the way, there are multiple studies making the case already that we should be using COX inhibitors to treat cancer. Maybe not on their own, but they're definitely an important part of treating the cancer. Then there's another plethora of studies saying that another enzyme that uses PUFA to create inflammatory mediators, that pathway is known as the lipoxygenase pathway, also LOX, L-O-X. The COX, C-O-X, stands for cyclooxygenase. Multiple studies, studies about COX saying we should be using inhibitors for cancer. There are multiple studies with LOX inhibitors saying, wow, look at what we did with this animal trial, with cancer, they shrunk the tumor. We should be using LOX inhibitors for cancer. Well, aspirin is both a COX and LOX inhibitor, and so is vitamin E, but that's a separate topic. So anyway, so aspirin has a systemic anti-inflammatory effect. So that's one reason to use it. But PUFA can also degrade non-enzymatically, which means it can get attacked by reactive oxygen species and form various bad byproducts, most of which are aldehydes, very toxic. In fact, some of them are known to be carcinogenic directly. So just by eating a lot of PUFA, even if it gets oxidized properly, convert it to energy, assuming everything's fine, there's no blockage of glucose, which none of these happens actually. It does have these effects. But even if everything goes fine, eating sufficient amounts of PUFA, just because it's unstable at room temperature, especially in the body because it's a higher temperature, about above 90 degrees, it's much more prone to oxidative destruction. And all of the byproducts that are created are toxic. In fact, they're genotoxic, and some of them are mutagens as well. So just by that, you can get cancer, directly carcinogenic effect.
That's just going to just destroy one of the highway lanes. It's a car accident that just causes damage, and then that lane has to be closed off. There's too much debris.
Yeah. Yeah. Think of it as the lanes 2 and 3, their construction and repair requires energy, but also requires the proper repair of DNA, because these lanes are constructed by specific genes which express themselves and result in the creation of proteins, and each lane is a specific protein. So by administering PUFA, by eating PUFA, which converts into genotoxic and mutagenic substances and it damages these genes that are responsible for the buildup of the highway 2 and 3, in fact, you can get the damaging highway 1 as well. And that's when you really get the rapid death. Basically, all of the 3 highways lanes are destroyed. So, PUFA is really bad if you eat it because its side effects are really bad. But just pure oxidation of fat, higher relative to glucose, it basically, because of the Rendell cycle, it puts the cell into a reductive stress. And whenever there is an overabundance of NADH, which is the main energy carrier molecule in the body will try to reduce that excess by using pyruvate as the emergency oxidant, and that's going to result in the buildup of lactate. And we do see that in metabolic diseases such as diabetes, both type 1 and type 2. These people oxidize primarily fats. They have a higher level of lactate than normal, than optimal. And in fact, doing things that can limit the oxidation of fat and promote the oxidation of glucose and lower lactate are known to be therapeutic. There's a drug that was synthesized in the former Latvian SSR. The drug is known as meldonium, also goes by the name mildronate. And what mildronate does is inhibits the excessive oxidation of fatty acids into the mitochondria. So because the cell can only oxidize either glucose or fatty acids, and let's say when the cell is stressed, it oxidizes mostly fat at the expense of glucose, right? If you lower that excessive oxidation of fat, then the cell has no choice but to start oxidizing glucose. That simple step, right? Just all you have to do is give the cell what it needs, and it actually prefers glucose.
And that's why lowering dietary fat sometimes can be very helpful for people relearning how to utilize dietary glucose.
Absolutely, yeah. And that's why losing weight in patients with diabetes type 2 is known to actually cure the condition. So how can it be a sugar-driven condition, considering that it's the loss of fat that cures it, and actually the gaining of fat that causes it? It's already known. And corroborated both by the weight gain and the weight loss experiments, and also by this drug, which was designed to inhibit the oxidation of the excessive oxidation of fats. And it was developed as a heart drug. So they have another corroboration that heart disease may be metabolic in origin, also caused by over-oxidation of fats. So this drug was developed as heart disease, but then they found out it works for diabetes. And then they found out that it also has a very potent doping effect on muscles. So in other words, your muscles also can do better if they oxidize more glucose than fat. And it was recently banned by WADA, World Anti-Doping Association. And in fact, the famous tennis player Sharapova got a 2-year ban because she was caught taking it. But it's a purely metabolic drug. So all it does, it limits the excessive oxidation, not the supply, the excessive oxidation of fat once the fat is already into the cell. And it kind of tells the cell, okay, now you kind of have to start oxidizing glucose. There's no other fuel available. So since we brought fat into the subject, niacinamide actually has a very potent anti-lipolytic effect. In other words, you can the cell can get fat from two sources. One is from the diet. Two is from the fatty stores that you have, which gets released into the bloodstream whenever you're under stress. Either way, if you limit the supply of fat to the cell, you don't have to worry so much about the oxidation of fat later on, because if more glucose gets into the cell relative to fat, then the cell is going to oxidize glucose. So there are two ways to attack the problem. And by the way, they're synergistic. One, inhibit the oxidation of fat already into the cell, and it's known to be high in diabetes, cancer, heart disease, very old and sick people, etc., because they all benefit from getting drugs that inhibit the oxidation of fat. And the second thing you can do is limit the supply of fat to the cell. How do you do it? Well, less dietary fat, right? And also do things that, first of all, limit stress, because anytime you're stressed, this causes the release of fat into the bloodstream. So limit stress, but if you cannot limit stress, take things or do things that inhibit the release of fat from your fatty stores into the bloodstream. And niacinamide happens to be one such substance through its conversion into NAD+. NAD+ has a potent anti-lipolytic effect. Biotin also has such an effect, and aspirin also has such an effect. So if we assume that a cancer cell is nothing but a metabolically deranged cell, which is over-oxidizing fat at the expense of glucose, and creates a lot of lactate, right? So the pathways that we've already addressed is pyruvate dehydrogenase, the NAD+ to the NADH ratio, right? Biotin also helps to bypass a little bit a blocked pyruvate dehydrogenase through an enzyme called pyruvate decarboxylase. And now we have aspirin, which first of all has anti-inflammatory effects. We already said that those are beneficial for cancer. Multiple studies have already called. In fact, there's some clinical trials that are already using COX inhibitors, not on their own, as part of the traditional cancer therapy, but they already have a recognized role in cancer. So aspirin is an anti-inflammatory traditional role. Nobody's disputing that. But what is less known is that aspirin is also an anti-lipolytic, so it's going to limit the supply of fat to the cell. And then it also has, at higher doses, it has an effect very similar to mildronate. It's going to directly inhibit the excessive oxidation of fat in the cell. So really covering all pathways here. I mean, at least the ones that are known. You could probably do even better if you add an oxidizing agent such as coenzyme Q10, methylene blue, the vitamin K, tetracyclines. By the way, tetracycline family antibiotics, unique among the antibiotics, have a known anti-cancer effect. There was a recent study with triple-negative breast cancer, a disease that's largely untreatable. I mean, they'll give you chemotherapy and radiation and surgery, but it has a very high mortality rate. And once it recurs, it's usually invariably lethal. So long story short, 200 milligrams of the tetracycline antibiotic known as doxycycline. And the word cycline should tell you that, because basically there's multiple cycles, rings into the molecule. And if you look at doxycycline, tetracycline, vitamin K, coenzyme Q10, they're all essentially the same molecule. The only difference is the number of rings they have in the molecule. But their key determining feature is that they can accept 2 electrons, all of them.
They can serve as that emergency taxi.
As an emergency oxygen, basically, which is the taxi in this case. So the carrier, the acceptor of passengers is the taxi on the highway. The acceptor of electrons in our body, ideally, is oxygen.
Yeah.
And if there's no oxygen, if there's no taxis, in other words, you can send some kind of other emergency vehicle that does not depend on the lanes, maybe a plane or something, or a helicopter, to pick up all these passengers that are stuck in the highway because of the roadblock. Nothing can pass through with wheels. So you can maybe send something, can take these passengers directly, ferry them over to their final destination point, and drop them off there. And that's what the oxidizing agents do. So, but I thought that, by adding, so we have a good, I think, mechanism with aspirin so far and the B vitamins. We're addressing all the metabolic roadblocks, at least the ones that are known in cancer. We can probably do it better. I haven't tried it yet by adding an oxidizing agent. Maybe that will be the next step, but K2, I try to keep it simple. It's like Occam's razor. If something works, then keep it to the minimum number of steps that are needed. It will be an interesting experiment to try to add an oxidizing agent, but I switched to another lane. I said, okay, so let's look at how aspirin works. Well, it acidifies the cell. I remember talking to Dr. Pete maybe 4 or 5 years ago. There was a discussion about cancer, and he said the body has a mechanism. He was criticizing chemotherapy because the chemotherapy tries to kill the cell. So does radiation, right? Aside from killing indiscriminately, right? He's saying, that's a stupid idea. The body can actually kill the cells if needed through a process called apoptosis. But the problem is that in order for apoptosis to work, the cell needs to be in a relatively oxidized state, which is another way of saying in a relatively acidic state. So he said the cancer cell protects itself from apoptosis by being in a very reduced state, which means alkaline, right? And if you can do something to drop the pH, in other words, acidify the cell, if the cell is sufficiently deranged that it's damaged, that the structural damage cannot be repaired because it can get to that point, it still doesn't make it alien cell. It's still your cell. But some cells, let's say, if it's too deranged, it knows it needs to go, and it will go if you let it. But the way the cancer cells survive is by building up these reducing equivalents, known as one of the most famous ones is N-acetylcysteine, a very popular supplement. Unfortunately, the main defense mechanism of the cancer cell against apoptosis. In fact, if you go to, God forbid you get cancer, you go to get chemotherapy or radiation, they'll specifically ask you, are you taking NAC? And if you say yes, they'll say stop it and go away and stop it for a week and then come back, because they know that if you take NAC, it's going to protect the cell from the cytotoxic effects of chemotherapy and radiation, which are really trying to do two things: either kill the cell directly or damage it to the point where the cell commits apoptosis. But if you take NAC, which is already pretty high in the cancer cell, then this process will largely be prevented. So in other words, the chemotherapy and radiation will not be effective. So I said, okay, so is there a way to acidify the cell? Well, one way is to get rid of this excess NAC because it's a reductant itself. So what can you do? Well, NAC and also reduce glutathione. I forgot to say that's the other major defense mechanism of the cancer cell. So NAC you can do by not consuming too many muscle meats because the cysteine is mostly present in them, which is the precursor to NAC. But what about glutathione? Well, glutathione, just like NADH and NAD plus and NADH, it has a redox pair. It has an oxidized version called glutathione disulfide, which is abbreviated GSSG. So GSH is the reduced version, GSSG is the oxidized version. And just like the NAD plus and NADH in a cancer cell, you have predominantly the reduced version, which is GSH, and insufficient GSSG. But they can interconvert into each other. All you need to do is administer an electron acceptor. In other words, it's going to take the hydrogen from GSH, so it'll be only GS, and get converted to GSSG. So I've tried that through another study, which was done in Bulgaria. Just trying to administer something that can oxidize GSH back into GSSG. And by itself, it didn't cure the cancer, but it made the chemotherapy that was used traditionally for that tumor, it made it much more effective. So I said, okay, so we're on the right path here. Dr. Pete was probably right. So what else can we do? Can we drop the pH of the cell directly? So one way is metabolically by administering oxidizing agents, which are acidic. And that's why carbon dioxide is so important for the health of the cell, because it itself is something known as Lewis acid. It's an electron withdrawing agent. So in other words, when your body operates properly, it produces the things that protect it from cancer and virtually all other diseases. You keep it. So by oxidizing more, you kind of increase your ability to stay in an oxidized state. And when you get into the reduced state, this promotes even more reduction through the various blocks which prevent the synthesis of carbon dioxide. So back to the question, can we acidify the cell directly? Can we drop the pH of a cancer cell? Well, the obvious answer is you should be able to if you can deliver something very acidic to the cell. Well, aspirin is acidic, but not sufficiently, or at least not in the dosage that most people associate with it. And I talked to Dr. Pete about it. He said the reason aspirin works for cancer in the higher doses is because it acidifies the cell. So I started looking around for molecules that look like aspirin, but are hopefully more acidic and also hopefully more lipophilic because their uptake into the cell is higher. And then I found by accident, because I was looking at basically more acidic analogs of aspirin, and I found a study from the 1940s, I think, which used something called 2,6-dihydroxybenzoic acid, and that's basically just the salicylic acid molecule is 2-hydroxybenzoic acid. So the only difference between salicylic acid and this new molecule is an extra hydroxyl group on position 6. In fact, it happens to be a metabolite of aspirin. So I found this study from the 1940s, which said that, and the rationale was the same. They said, we want to use something. We've been giving patients with rheumatoid arthritis, we've been giving them 8 to 10 grams aspirin daily. There's some side effects there. Basically, they have like stomach upset, prone to more bleeding, right? What else? Headaches. Sometimes they get headaches. Their temperature rises because aspirin is actually a metabolic uncoupler. So people find this, uncomfortable. They cannot sleep at night. So what else can we do? So they basically tried this metabolite of aspirin, and I don't know how they managed to choose that specific one, but they said, oh, here's a metabolite of aspirin that is much more acidic and much more lipophilic. So let's see what happens when they give it to patients with rheumatoid arthritis. So they gave it at a dose that was 1/10 of the dose of the aspirin that they gave to the rheumatoid arthritis patients. So in other words, if the patients were getting 8 to 10 grams of aspirin daily, they gave them the equivalent of about 800 milligrams of the 2,6-dihydroxybenzoic acid. Wiped out their RA in 24 hours. No complete disappearance of swelling, immediate reduction of the fever, normalization of the blood parameters. And then basically, to me, it was, okay, that's it. And then I started searching more for this molecule. And then I found a Google patent which proposed combining aspirin with some more acidic molecules, and it listed about 20 of them, but one of them was 2,6-dihydroxybenzoic acid. And the patent says the combination of aspirin with something that's more acidic than aspirin should be able to treat cancer. And it's just a patent. It doesn't give you a study or anything like that, but it's a claim. Somebody thought that this could work. So I said, okay, let's try it. So I basically did another experiment where I had the 3 groups. One with the aspirin and B vitamins regimen, right? And then one with the 2,6-dihydroxybenzoic acid at a human equivalent dosage of about, 50 milligrams. 30 milligrams will be about 200 milligrams daily for a person, and the B vitamins. And then I had the 2,6-dihydroxybenzoic acid by itself. So the 3 groups were B vitamins, aspirin at the dosage used before, B vitamins, 2,6-dihydroxybenzoic acid, no aspirin, and the dosage for the 2,6-dihydroxybenzoic acid was about 200 milligrams daily for a person, and then 2,6-dihydroxybenzoic acid by itself. All 3 groups cured, but the ones that had the 2,6-dihydroxybenzoic acid had a much steeper curve of regression of the tumor than the one with aspirin. So I said, wow, this works. And then I started doing more experiments with 2,6-dihydroxybenzoic acid at a higher dose. Then I also tried to do a higher dose of aspirin combined with the B vitamins. And then I tried to combine 2,6-dihydroxybenzoic acid with niacinamide. So 2,6-dihydroxybenzoic acid at a dosage of a human equivalent of about 800 milligrams that they were giving to the animals also cured it, but it was not as effective as in the group that had half that dose, which was only 50 milligrams per kilogram for a mouse, which is about 400 for a human. And then the B vitamins plus aspirin at a higher dose worked worse than the B vitamins plus aspirin at the lower dose, the dose that's been tried in human trials for Crohn's disease and for multiple sclerosis. So now basically what I'm trying to do, so I know that the B vitamins plus aspirin at the dosage of the dose that we already described, it's already been tested 3 times. All 3 times they've been cures. So now the goal is to find out the optimal dosage for 2,6-dihydroxybenzoic acid, because it looks like it works best when given on its own. When given in the combination with the B vitamins, it still regresses the tumor, but not as steeply as basically the B vitamins plus the aspirin. When it was given together with aspirin, as in that patent, it did not work. There was one regression, but partial, and 2 of the animals died. So that patent failed, but it was good that it put me on the right track to find that molecule. So right now, trying to come down to basically two types of treatment. The B vitamins plus aspirin, already proven 3 times. 2,6-dihydroxybenzoic acid by itself, likely the dosage equivalent to about 400 milligrams daily for a person. And also now trying to see if 2,6-dihydroxybenzoic acid with niacinamide would work even better. There was some indication that it might.
Why do you think that it didn't work as well with the B vitamins present?
I have no idea. I mean, that something needs to be teased out. There is a potential that it could acidify the cell too much, in which case you get into something called true oxidative stress. In a very in an organism that's in a very precarious health state, sometimes when the cell is too stressed and you very rapidly shift it back into oxidation, it can die. Yeah. So basically, it's kind of like the refeeding experiments of did you say the Wisconsin starvation experiment?
Minnesota starvation experiment,
yeah.
MSC.
Didn't they do the refeeding and they found a lot of people died when they refed them too quickly?
Oh, they had not in the MSC, but
okay.
they stated literature saying like how much, like for example, the low pulse rate, when they refed quickly, their pulse rate skyrocketed.
Worse than that, look up something called the refeeding syndrome. I think they did it with Holocaust survivors. If you take somebody who's been starved for too long and you immediately put them on a regular calorie diet, they'll die.
Yeah.
So that's my guess for the cancer cell as well. Basically,
is that probably why the higher dose didn't work?
Exactly.
Too?
Yeah, that's my guess so far. I mean, obviously, it can be investigated more, but right now the goal is to get to two treatments. And then start testing them on other models. So I'm on the last study that's basically going to determine what's the optimal dose for 2,6-dihydroxybenzoic acid. And in that last study, I'm also testing aspirin at a much higher dose, at a dose that Dr. Pete said should treat the tumor. I tested it at a low dose that's used for multiple sclerosis and Crohn's disease. I tested it in intermediate dosage. That did not work very well. It actually worked worse than the lower dose. But now I'm testing at a dose where Dr. Pete said it should be able to be used as treatment for
Georgi,
cancer.
I know you have unlimited money and unlimited time, but could you?
Not true.
Is there a way
no,
to do future studies where you keep once you find the B vitamin and aspirin or this other molecule's dosage that you like, is there a way to then modulate the macronutrient and calorie intake of the mice to see what the impact of food as well?
Well, I already did an experiment for that. I wanted to check what can a fat-free diet do for the tumor. So basically, I tell them put the mice on a fat-free diet for 2 weeks. I think was the period, and then try to inject them with the tumor. And they put them on the fat-free diet. The mice, they did a blood test. The mice had a essential fatty acid deficiency, which kind of confirms that they've been depleted of the polyunsaturated fatty acids. And then the next step usually is when you try to do a cancer study, is they take the human tumor, they destroy the immune system of the mice. I think they do radiation of the thymus gland or something. But basically they immunocompromise because otherwise they'll reject the tumor. It's not mice tumor, it's a human tumor. So they destroy their immune system and they try to implant the tumor. And usually it works with one injection. These mice that were depleted of the polyunsaturated fats refused to hold a tumor. They could not be inoculated.
Wow.
So they tried for 2 weeks, and eventually we decided basically like we'll give up. They said something is going on with these mice. We've never seen this before. Confirmation of Ray's statements that essential fatty acid deficiency makes the organism extremely resilient to a variety of toxins, and an inoculation with a cancer cell line, I think, classifies as a toxin. I will try to vary the macronutrients. I will try a fully saturated fat diet, right? 0 PUFA, just because we need some diet there to see if it's better than the fat-free diet. My guess is that the fat-free diet, but it's difficult to test because you cannot give these mice tumors. So I don't know what we'll be testing there. Maybe with the saturated fat, we'll develop. Maybe I'll first inoculate them with a tumor and then put them on the fat-free diet and the saturated diet. Mm-hmm. That's the other experiment. So that's a future project for me.
Oh, please keep us posted.
Yeah,
I will.
I think that our audience needs to remember though that like you said, sometimes more is not better.
No,
so
definitely.
Like, if we can find the minimum effective dose, that's usually the best route forward. Because I think a lot of people listening would probably go and jump to taking like as much of these supplements as possible if they were really learned helplessness.
Remember,
and freaked out.
There's a difference between prevention and cancer treatment.
Correct? Yes. Yeah. And also, as Ray said, basically when somebody asked him like, well, if somebody's hypothyroid wouldn't take such a dosage, I forgot what the molecule was. Wouldn't that be dangerous? And Ray said everything is dangerous when you're hypothyroid, so you got to take it slow. Initially, you got to get to a point where basically you can tolerate things. If everything irritates you around you, if you shake all the time, if you lash out at the slightest, I don't know, provocation, chances are that you know, in a state where you cannot really take that much more stress. And a drastic change, even though for the better, is also stress, just like the refeeding experiments.
Yeah, that's true.
Yeah. Do you have anything?
No, this has been very fascinating. I've loved listening to this.
Yeah, so just to reiterate, 2,6-dihydroxybenzoic acid, it's not some kind of an esoteric exotic chemical. It's a metabolite of aspirin naturally forming in the body, though in much lower amounts, even if you consume large amounts of aspirin, than what you will do with direct administration. However, it may very well turn out to be the main mechanism how aspirin works for cancer through its metabolizing into that substance, among others. It has other metabolites as well.
And it's helping restore proper glucose metabolism instead of killing your cells through radiation.
It actually acidifies the cell, and basically if the cell is sufficiently compromised structurally, it will commit apoptosis because
apoptosis naturally.
The rest, for the others, because it's also a highly pro-metabolic substance, it inhibits the oxidation of fat. Then the rest of the cells that are cancerous, but they're not too damaged, not too deranged, then they're getting the glucose from the bloodstream and slowly reverting to normal.
Oh, okay. So it's kind of like treating cancer at two levels. It's filtering through, okay, these cells may be able to, they're not too damaged yet.
Yeah.
So let's repair glucose metabolism, and then let's acidify the other cells that are too far gone and they just need to die.
In a miraculous hit of coincidence, there was exactly such a study with breast cancer and aspirin, which I posted on the forum now with a different name, maybe 4 or 5 years ago. I didn't, it's really coincidence because the title says aspirin can cure breast cancer and by making them revert to normal, and kills the ones that refuse to revert to normal.
And the huge difference here is that their body is able to select which ones can be killed, whereas with radiation, you're killing everything
indiscriminately. Path. And there's also the so-called bystander effect. So when somebody gets a dose of radiation, a very common side effect is they get flushing and redness and inflammation throughout the entire body. Why? Because the cells that were killed, they undergo something called necrotic cell death. They disintegrate and spill literally the entrails into the bloodstream. This is already known to be able to cause a variety of very bad conditions, including autoimmune diseases, because the mitochondrial debris and fragments and various other ones that we probably even don't know about are capable of activating certain receptors, such as the endotoxin receptor, now known to be behind many diseases. They're also capable of activating the inflammatory pathways, the COX and the LOX pathways. So you're going to start producing more inflammatory mediators. So the body treats cellular debris into the bloodstream the exact same way as it treats a bacterial infection. Which kind of corroborates the old view that really the human cell is a symbiosis between, a primitive cell that could only do glycolysis, which is the cancer cell, and then a bacteria went inside and they formed a symbiotic relationship, and the bacteria became the mitochondria, because the mitochondria has its own genome and it's different from the cellular genome. So it does look like it's a different organism, but and that may explain why people, when they're under stress chronically, they can develop autoimmune conditions. Because when you're constantly under stress and you have cellular disintegration because of the stress hormones, because that's what they do, they kill you. Then they don't kill; they shred your fat cells and the muscle cells and the bone cells and many others. So the proteins in that cell go to the liver to get converted for glucose, but everything else that floats around basically activates a variety of inflammatory pathways, and at the very least, you'll be under chronic inflammation. And over time, the body probably adapts and says, well, that the place where this debris is coming from is diseased, right? And I need to basically get rid of those sick cells. So it starts producing antibodies for the specific cells of that specific organ. There are multiple studies now that show that giving cortisol actually increases inflammation in the long run. So it's a really very pernicious and perverse and ironic treatment. You go to the doctor with high inflammation, let's say for autoimmune condition, the doctor will put you on cortisol. And while you're taking the cortisol, the inflammation is subdued. But in the meantime, behind the scenes, cortisol upregulates all of the inflammatory pathways that we know about: the COX, the LOX, the NFKB, tumor necrosis factor alpha. So as soon as you stop the cortisol, the synthetic cortisol, your inflammation rebounds to a level 2 or 3 times higher than baseline, higher than what you had before you start taking the cortisol, which could explain why after people start on glucocorticoid therapy, they seem to never be able to wean off because they develop progressively high inflammation. Then it's higher glucocorticoids to tame it down. Then they develop even higher one, and it's a vicious cycle. So you need to do something that lowers inflammation, but not through the glucocorticoid pathway. Cortisol's purpose, the main one, is to keep you alive by providing supply of energy, which is coming from you. It's definitely not a beneficial pathway. It was not meant to be activated all the time.
It's necessary in small doses, and it's okay. But
acute. Well, I wouldn't say small doses. Acute and intense to run away from the saber tiger, that it was chasing you when we were evolving, back in the savannah, right? But after that, it should subside. If it doesn't, the sign is that you're under constant danger. In a situation like that, the body will say, okay, keep cortisol up, get rid of everything that's not necessary for survival. And it might sound like a good thing, but it really deprives you of everything that makes you human. It will kill cognitive function. You'll kill mood. Cortisol is a known depressant. In fact, some of the most successful antidepressants are cortisol antagonists. You'll kill libido. It'll destroy your muscles. It'll actually make you put on fat, because the signal to the body is that if you're under stress, the body says, I should be very careful and stingy with my production of energy. I should store most of what you eat instead of burning it right now, because you're definitely under stress, so which means I need more reserves. So cortisol will make you fat, while simultaneously destroying your lean tissue. So you're going to get eventually something called sarcopenic obesity, which is just a scientific term for something known as skinny fat. So basically, your limbs are very thin. You have no muscle whatsoever, but you have a bulging midsection.
Oh yeah. Well, I was gonna say, like, that you said, like, that doesn't make sense to take that, but treatment plan wise, it goes with your conspiracy theory that it would make sense to continue to take this drug, and then they have to go back for more and more treatment.
Well, Goldman Sachs certainly agrees with me. They didn't call it conspiracy. They said short that stock. This company deserves to die because it's a very poor business model to get rid of your clients.
And autoimmune, the way that mainstream views autoimmune is very similar to cancer, where it's like, this is a foreign thing we got to get rid of.
Attack itself.
And actually, one of the reason cancer therapy developed like that is that they actually were looking at autoimmune conditions and saying, well, if the body can attack its own tissue, clearly it makes sense for us to create this theory that says, well, there's, you can get like the ultimate of example of alien tissue with cancer. So a more mild version will be autoimmune condition, and we know that the body attacks this tissue. So the public is going to be able to accept the idea that in cancer, which is really, really alien cell, we have to kill that cell. The body cannot kill itself, or it's going to kill you. And it's really like a spectrum of it's viewing the world as inherently evil, including your own body. So you can definitely turn on yourself as far as medicine is concerned.
Huh. Oh. Okay. Well, I think
can I ask a question?
Yeah,
okay.
Of course.
So outside of your self-experimentation for the sake of Idea Labs, are there any of these vitamins or compounds that you personally take regular as a preventative measure?
Mm-hmm. I take aspirin preventively. I used to take it daily, but then I asked Ray, I said, what about, for cancer, cardiovascular disease, dementia, all the things associated with aging. I'm not exactly a young buck anymore. And he said, well, I don't think you need to take it daily. I said, how come? He's like, no, aspirin has a very long half-life, because when you metabolize into salicylic acid, the half-life is like 34 hours or something, which means if you take it every day, it's actually going to start to build up, and you don't want that. And then he referred to some studies showing that if you take it once a week, that's sufficient for cancer prevention. And then I dug more into that, and I found studies that saying that for some cancers, even twice a month is enough. That's all that you need to take. Probably because it builds up, or because it fixes some of these metabolic derangements that are there. So I take twice a week. I take a regular tablet twice a week. I take niacinamide at 500 milligrams daily. And maybe once a week, I take vitamin D and K. Everything else is food.
Yeah.
Awesome.
Food comes first. I think that's the most important thing, guys. Like, make sure you're hitting your bases. You can plug these in a cronometer and just make sure like you're hitting your RDAs in food.
Yeah.
And then see how you're doing and add supplements as needed on top of that.
And read the label very carefully. Even most of the organic foods, they contain things like carrageenan, which is now accepted as a known, proven human carcinogen in many countries. US is not one of them, even though not that US is evil, but there have many doctors that petition the FDA to label it as a carcinogen, and FDA says it's not sufficient evidence. But the evidence is there. Whether it's sufficient or not, it's up to you, not up to the FDA.
Yeah.
So it's a known carcinogen. Most of the gums, like acacia gum, guar gum, locust bean gum, all of these gums, they have known inflammatory effect in your gastrointestinal tract. Anything that irritates your gut leads to chronic inflammation. Anything that leads to chronic inflammation inhibits oxidative phosphorylation. Anything that inhibits oxidative phosphorylation, ultimately, in the words of Otto Warburg, not mine, will cause cancer. And probably other diseases before that, like on the spectrum. You'll probably get first diabetes or cardiovascular disease or something, and eventually, if it doesn't get treated or like doesn't get mitigated, you'll get cancer.
I think that spectrum is important to understand, because then it doesn't allow you to, compartmentalize yourself in a specific, like, you got to go through this door and now I'm labeled here, I'm stuck here. Yep. It's like a spectrum, and you can move yourself back and forth.
Forget about the named diseases that medicine has. There's no such thing. I mean, sure, you can have a specific derangement in a specific organ, but all that means is that that organ was the most energetically compromised, so the disease manifests there. All disease, until proven to the contrary, should be considered systemic. It may have organ-specific manifestations, but it's always systemic. So if you have like a liver problem and you go to the doctor and maybe they do something for the liver, that often does not fix the problem, and the problem recurs because it's a systemic problem. You have a systemic energetic deficiency. The liver just happened to be the most compromised because probably of your lifestyle or whatever you had it from your parents, non-genetically, because people say, oh, look, genes work. Not in the way people think. There's no gene, despite 100 plus years of looking for a gene for a specific disease, none has been found. Then they started looking for groups of genes, said, okay, maybe it's not one, maybe it's 10 genes. 0 results. Then they started looking for so-called gene-wide association studies, which basically look at how genes work together, all of the genome, still failed. I mean, they have some results saying that, specific interaction of the entire genome in a specific configuration is associated with autism. But then they tried in animal trials to give drugs that silence or activate some of the genes that they thought were important. The animals got worse. They didn't cure the autism. So it's not genetic. I mean, very few, unless you are born with this, as Ray said, unless it was present at birth, even then, because you can inherit things non-genomically from your parents, epigenetically, unless it was present at birth, everything else should be considered metabolic, preventable, and above all, curable.
And that should be very empowering and freeing. So, Georgie, we got to run.
Okay. How's the farm?
Oh my gosh, so much is happening. New calves arrived yesterday. We have a small scale farm here. We have so many farm partners. And I think that's been one of the most fulfilling things I've ever done, is giving them a market so that we can bring back small scale farming. Yep. None of them are big scale at all small scale regenerative farming, and I like that. Building a partnership of them. It's so fun. Mm-hmm. It's so fun. It's so fulfilling. It
puts you back in touch with nature. I think that's what we're missing in the city. And unfortunately, I don't see how it can be reversed in the city. I think people are starting to realize that the city model came from the 19th and early 20th century. So the city was a place where you put a lot of people because the factories were there, right? And now that the factories are no longer there, most of the Western world does not produce much in the actual city, then the very purpose of the city kind of goes away. And even when they were there for purposeful activity, cities were not known for, for a place where you go and meet healthy people. You go to London in the, 1800s, or like, many of the European cities or even American ones in the early 20th century, cities were where you go and find the sickest people. It's the people that are, outside of the cities that usually live the longest and were healthy throughout their lifespan.
I get stressed when I walk into a city.
Yeah.
After being out of it for so long. Yeah.
You know the Dunbar number? Have you heard of it? So there's this sociologist or anthropologist, maybe both. So he basically said, in primitive societies, if you look at the average size of a tribe, it's about 150 people, very rarely above 200. So he says, our brains have evolved to form close personal relationships with no more than 150 people. In fact, with most of them, they're acquaintances. And then in a factor of 3 further down, so 50 people will probably be your extended family. Divide by 3 again, you get about 15. That's probably your immediate family. And then divide by 3 again, and it's basically you, your partner, and your kids. So he's saying, so it's like layers of up to 150 where your brain can actually comprehend these people and feel related to them and connected to them, so you don't kill each other over stupid stuff. But he's saying in the city, all of this is violated. So basically, like most of the people around you are strangers, and inherently, he's saying evolutionary, it activates a stress response because a stranger is always a potential danger. Don't they tell the kids stranger danger? Like
Stranger
whenever they
danger.
put, yeah, exactly.
Wait, and then now add social media on top
All of
of
it.
that.
Yeah, that's
where it's
Constant
billions.
bombardment with EMF, like with basically crappy food, like traffic, which is another thing. So basically, not only you're surrounded by strangers, they're acting in a very threatening manner most of the time when you're on traffic. You have to be always on your guard, on alert when you drive in the city. You never know where it's something, shit will materialize out of nowhere. So you have to be, really it's very stressful to be in the city. And even when you go home, of course, there's the routers and everything else that's in the city. I apologize again. I guess it's the office way of saying enough of you blathering.
No.
So yes,
Yeah.
I think cities are inherently stressful. When you're young, it's stimulating, but it's not a coincidence that many people, when they get a little older, like especially if they pair up, they basically leave the city and they go to the suburbs.
Yeah.
You just can't take it with the added stress of having family. Because family is probably the most meaningful thing you can have, but it's also one of the most stressful experiences in life.
Yeah. How are your kids doing?
Fine, in school. That's why I'm talking to you, because they're in school.
Well,
If they were home, I wouldn't be able to do it during the day.
we should have the last podcast we talked about, talking about what your kids are kind of being exposed to,
Yeah.
and navigating that type of thing. I think is a very interesting conversation, given like the next generation is experiencing challenges that we didn't experience, right? And so now that's adding another layer of stressors into their metabolic environment that I think is going to have profound impacts on their health in 20, 30, 40 years. That
I think social media has to be somehow handicapped at the knees, or it will consume society and destroy it. It's just very few positive things that I've seen come out of social media. Some people say, well, society changed and used to be good and now it's bad, and that's why social media just reflects that. Yeah, but social media wasn't an innocent bystander. It contributed to that, right? You hear some of the most vile and extreme voices on social media, and those tend to be amplified by the algorithms, which, by the way, want your eyeballs so they can sell you ads. So it's very similar to the pharmaceutical industry. The pharmaceutical industry wants you sick but not dying, not until they've made enough money off of you. And the same thing with social media. It wants you stuck there, right? And of course, the medicine likes social media because it makes you mentally sick. That's another reason to prescribe you drugs. And then it's already been admitted that it's addictive, right? Well, if you have no problem banning addictive drugs, why do you have problem? I wouldn't ban it because then it's going to create the forbidden fruit dilemma, right? Yeah. But there should be a way to drastically scale down social media usage. And it's not by banning things, but I think by providing more opportunity for people to have personal connections, which unfortunately, in my opinion, can happen mostly outside of the city. It's just too many unknown people, and the modern cities tends to combine some of the worst features of humanity, and for a very good reason. I mean, like you go to a large city to be predatory, to exploit other people, to make money, which is kind of like what the big cities are advertising. Greed, lust, individualism. I was watching this. There's a movie about the first nuclear Russian submarine, and they have this situation where the political officer, the KGB guy, who's of course member of the party, is showing these movies to the sailors. They're all communists, right? And he's showing them basically how in the big cities everybody has a nice car, nice apartment, and et cetera, but you never see the sickness and the vile whatever behavior behind those lies. He says, remember, the city propaganda appeals to your three most basic instincts: greed, lust, individualism. I don't have a problem with individualism that much, but like when everything is about greed and lust, and think about it, most of the modern culture is about that, right? Greed associated with money, and lust, sex. Money and sex, I think, is that if you can sum up a Western city in 2 words, I think it'll be those.
Yeah.
I'm stressed talking about this.
Yeah. Okay, Georgi.
Yes. Go distress.
We'll get you back on the schedule, and I can't wait to hear about the next round of cancer studies. I think that this is really fascinating, and I really want everyone to appreciate that Georgi is doing this with his own money and all of his unlimited free time that he has. And so, Georgi, know that I am very grateful for your endeavors.
Thanks
here.
For making me laugh. Unlimited money and unlimited free time. Not.
So, where can people find you?
Support
Idea
you.
Labs, what are you?
Just to
any
answer
new
one question,
offerings there?
you asked me at the very beginning. The lab is basically, it's an American company. The company is formed here, but the lab is actually in Taiwan.
Okay.
So the way this works is basically I sign a contract with they're a licensed pharmaceutical company. In fact, they make drugs as well, but they also run clinical research.
Cool.
Their clients are Pfizer. It's a legit lab. In fact, they wouldn't do nefarious things, right? And all the work is done by them. All I do is I provide some links with studies and say, here is the rationale. Here are the compounds I want to test. Give me X number of animals. This is a dosage that I want to try, and they take it from there. So there's no participation in my way, in any way, on my side, in any way they can actually, I don't know, corrupt the research.
Yeah.
They have a name to protect, so all the work is done by them. I just get a report. I can send you one of the reports to look at, where basically it's a spreadsheet, and by each period of 2 or 3 days, they measure the tumor, they measure the vital signs of the animal, etc., and then they give you a graph for each day, like basically it's a plot where each dot is separated by 2 or 3 days, and you can see the tumor growth. And eventually, if the animals die, they do a necropsy, they tell you what the reason was, etc., So that's really how it works.
This is like the ultimate way to honor Dr. Ray Peat, and I think it's really beautiful. Is there a way that people can help fund these studies?
I'm getting questions about this. Once I'm still in the shooting, not so much in the dark anymore, but once I settle on 2 or 3 treatments that I think work, that have shown promise, right? Then I'll try it on several other xenograft tumors.
Okay.
And after that, what we can do is a crowd-funded clinical study under the compassionate label by the FDA. So this means is some people that are terminal, been declared terminal by their doctor, they have nothing to lose, right? So they sign a disclaimer saying, okay, I'm going to take these things. I'm not going to sue Georgi or anybody else who is sponsoring this. But you still have to pay them. They need to be under the care of the doctor, right? So somebody needs to cover those expenses. So after the animal studies are done, I think the next step will be to do a crowd-funded human study with, I don't know, 10, 15 people. And then I think will be the best time for people to contribute because it'll cost a lot more than the mouse study.
Okay, you should get a link set up on Idea Labs in preparation for that.
Okay, I will.
That'll be really cool.
Where can people find your blog?
Oh, it's HAI D as in dog, U T as in Tom.me. So Heydut, which is my online alias, .me. And then that's about it. I mean, I used to post on a forum, but not anymore. I could change the name and change the direction.
Forum? What is
Low Toxin Forum, yeah. And by the way, when they changed the name, they broke all of my links. So now that all the Repeat forum links that I've been sending to people, when they click on that, I think it shows them like it should have just created like a forwarding thing, right?
Yeah.
But now it's broken and people are starting to email me and say like, well, what's going on? You sent me this stuff 2 years ago and I just wanted to read it again and it's gone. So unfortunately, the only way to get around this, actually 2 ways. One is to go to the forum and search for some of the keywords that are mentioned in the study. I mean, in my post. The other one is Brad Cohen. I think you know him. He created the Peatbot.com, the agent. Yeah. So he set up one alternative forum, and there's several other people setting up other forums that are more dedicated to the bioenergetic idea. He said he has a copy of the old forum. So basically just take maybe at least my posts because I own them and repost them on a new forum where they'll be, they'll be searchable. And
Georgi forum. Let's go. That's awesome.
No, bioenergetic.forum. He already formed it. But he'll take my posts and just repost them there as threads. Awesome. I don't care who the author is, but just so people can have access to what was posted in the past.
Cool.
Great.
And you guys can check out Georgi's supplement line, Idea Labs, and check out his blog posts, Heydut.me. And we're excited to have you back on again soon, Georgi. Appreciate your time.
Me too. Thanks for inviting me.